Welcome, everyone. I'm Ken Lucien, Dean of the College of Engineering, and welcome to the Future of Engineering Symposium, followed by the Alumni Awards ceremonies for the College of Engineering. This symposium was established in 2008. It was originally established as a tribute to Professor Merrill Ebner, one of the iconic leaders and figures in the history of the College of Engineering, highlighting the societal impact of engineering. And it's thought that we would do this over a range of disciplines. The first one being a more general presentation of all possible applications to society of engineering. The second one focusing on energy, and now this one, of course, focusing on personalized medicine. We call it personalized intelligent medicine, or how engineering, technology, innovation, and public policy challenges will impact healthcare in the future. Many of you know that there's been some extraordinary advances in engineering that have an impact on the way medicine might be practiced. They range from the biologics aspects of medicine, areas like genomics and synthetic biology and systems biology, and how that may alter the way we can treat these different diseases in medicine, which you'll hear from Jim Collins soon, and to the medical devices and technological breakthroughs which may fundamentally alter the way medicine is practiced through devices and home health care. But all of these advances, of course, can't be created and developed in isolation by engineers. They have to be developed in a very complex ecosystem of healthcare, economics, and public policy. And there are some major public policy issues associated with all of these technologies. And that's what we're going to hear about today. We have two keynote speakers. Our first speaker, you'll hear, each one will speak for a few minutes. And then about 20 minutes or so, if I remember what's the instructions. A few minutes got them nervous. And there really are two iconic figures in each of those areas. And then we will have an open discussion from the audience about issues that you would like to discuss with these experts. First speaker will be George Savage. He's the co-founder and chief medical officer of Proteus Biomedical. But far more important than that, he's an alumnus of Boston University, Engineering 81. He began founding technology-based medical companies in the Silicon Valley 20 years ago. After 7 years of practice as a physician specializing in trauma and emergency medicine, he got his start in the medical industry working for Mark Levin at the Mayfield Fund, assisting him in founding Cell Genesis and Abgenix. He then co-founded CardioRhythm and served as a director and vice president of clinical and regulatory affairs through its acquisition by Medtronic. Next, George co-founded FemRx and served as a director and senior vice president of research and development. During his tenure at FemRx, he completed an IPO and was subsequently acquired by Johnson & Johnson. Currently, he is co-founder and chief medical officer of Proteus Biomedical and also co-founder and managing member of Spring Ridge Ventures. His company, Proteus, is developing intelligent medicine products that address therapeutic areas where disease management complexity, patient monitoring requirements, therapeutic efficacy, and poor patient compliance are major issues. The company's ingestible technology makes intelligent pharmaceuticals possible in ways that provide patients and doctors with valuable information to customize their therapy. George will discuss in more detail how his technology has the potential to improve patient outcomes and impact the future of healthcare. In addition to his bachelor's degree in biomedical engineering from Boston University, he received his MD from Tufts University and his MBA from Stanford Graduate School of Business. I have to tell you, George is one of the first alumni I went to visit outside the Boston area as dean, and I was just blown away by George and by his technology, and I hope you have a similar reaction. Please join me in welcoming George Savage.
Well, thank you very much, Dean, for those introductory remarks. It's very kind. And let me try to get the technology to work here. Yeah, they tell you never perform with children, animals, or technology, and we're throwing that all to the wolves right here. Slideshow mode. There we go. All right, fantastic. I think I'm technology compliant now. It's a humbling and wonderful experience to come back as an old guy to my university here. A little weird, but it's really good fun. And thank you very much for the opportunity. What I'm going to do today is spend a few minutes, and I've got to check the time to make sure I don't rabbit on too long, talking about some serious problems in the medical industry, some real significant issues regarding innovation and the vector of innovation, then describing Proteus as a case study of one of the many myriad opportunities or solution sets being developed out there, and then conclude with how this really adds up to an enormous opportunity. You know, one of the things for all of us at every stage of our career is to realize that no matter how daunting the problem, you can flip that around, and the other side of the coin is that you're looking at an amazing opportunity. So we're going to have a little bit on the pharma industry, Proteus, and then what we're doing about it. But just a personal note about some of the developments we see here in medicine. Medicine is all about innovation. Innovation is something we really support and everybody loves. But I would submit that the vector of innovation in the medical industry, particularly pharmaceuticals, is in the wrong direction. We're over-investing in absolute therapeutic capability. The pendulum is swung too far away from the 19th century approach to medicine, which was highly personal but pretty useless, technically speaking. But your doctor knew all about your social situation and was there providing whatever placebo effect could be provided. And now we're at the other end, where we have proven therapies that work very well in statistically controlled populations in a very theoretical construct, but work much less well for actual patients. And with that, I'll take you through my disclosure. You already heard about me and who I am. The most important point is right there. What I'd like to open with is just a real issue about the problem facing the medical development industry, the pharmaceutical industry, and it really boils down to, on a worldwide basis, issues of access, value, and sustainability. And just a few factoids to illuminate the conversation and everything that follows. We spend an awful lot of money to achieve a higher level of outcomes in the West, but we often don't get everything we think we're getting for it. And just to take you through a few very signature technologies, the implantable cardioverter defibrillator, this device in your chest that monitors your heart rate and will save your life. In terms of implants per million, the U.S. leads overwhelmingly there. It's a very expensive technology. It's more than $50,000 skin to skin, buying the device, going through the surgery. 650 per million in the UK. It's a fraction of that, and they have a very controlled healthcare system, as you know, with 95. And in China, almost nobody gets this. If you think of lives saved per patient, Eric Topol, who heads now the West Wireless Life Science area, formerly at the Cleveland Clinic, has a very good talk, really elaborating on these themes at length. But if you take the most successful, the defibrillator, 7 out of 100 implants save your life, and the other 93 do nothing at all. Lipitor, it's a drug you're on for life, 1 in 1,000 additional lives saved. Mammograms, this is something we all like. My wife gets mammograms, I recommend them, but it's 7 per 10,000 mammograms, so very low yield. And if you think of productivity of research and development and where are we investing our marginal dollars, and correspondingly, are they going to the right place, the best use? Well, the evidence tells us that they aren't. The research dollars required to get a new molecular entity out of the pipeline to the market in the pharmaceutical industry has gone up by a factor of 10. Well, I'm sorry, not quite a factor of 10, a factor of 5, roughly, in 10 years. So significant changes there. So more money, I would submit, is not necessarily the solution in aggregate. The U.S. spends a lot more money than any place else on earth. Clearly, we enjoy a higher level of service. But in terms of the raw outcome, life expectancy, it really doesn't vary that much between the U.S. and other places. Just to give you a little more data on this, these are engineers, so I've added some slides here to be a little more data-driven. You can see from this graph here that pharma R&D spending has escalated dramatically, but the number of molecular entities coming out the pipe at the back end just hasn't budged at all. So we're spending more for more expensive therapies and getting less out of it. And I would submit that this underlies all of the political Sturm und Drang about healthcare reform. Are you for it? Are you against it? What does it look like? The reason people are fighting now in a way that we didn't used to fight before is that the current way of doing things isn't sustainable. And a theme I'm going to get into is rather than fighting over who gets the remaining deck chairs on the Titanic, we should try to innovate ourselves out of the box we're in. And that means not just technology innovation, but business model innovation and taking risks in terms of how you get paid and how you define a new and exciting therapy. This is just another example. Pharma companies are becoming uninvestable. Fewer and fewer compounds. It costs more and takes longer to get them out. If you want to do any research, pharma patents are expiring. And the drugs that are now commodity chemicals, generic drugs, are quite often very, very good for the diseases that they're used to treat. The problem for most patients is that they don't get the benefit that's theoretically available. So we have absolute therapeutic capability up here. Most people in the real world only get something down here, and that again leads to a lot of the dissatisfaction with the healthcare system because people don't really know what's going on or how to manage their healthcare. One of the problems is that this is what the chronic disease healthcare treatment system looks like to most patients. And you know, fortunately, I think most people in this room are too young to have any of these issues. Hopefully you won't for a very, very long time. But trying to manage a regimen of multiple drugs when you have multiple diseases is very problematic, and it's an information problem. When you are in the hospital, just to give you a contrast, the doctor knows what you're getting because there's an order and a nurse gives it to you. The doctor knows how your body is responding to that therapy and can make adjustments, the wrong medicine, the wrong dose. Then when you are better, you go home. In chronic disease, no one has any idea. Your family members want to help you. They don't know what's going on. The doctor wants to help you. He only has your report of what's going on, and patients don't report these things accurately because they're not machines. And so you're essentially throwing darts in a dark room. And then the final point to make in this vein is healthcare is the only thing left in life that I'm aware of that you can only handle in person. So I was in Europe visiting with a Proteus corporate partner a month ago. I realized to my astonishment I forgot to pay my Pacific gas and electric bill, and it was due that day. So I pulled out my phone and I paid it in just a couple of minutes. My mother, almost simultaneously, was having an issue that may require her to have some spine surgery. I spent days trying to reach the orthopedic surgeon on the phone, and finally I had to fly to Florida. I had to go to the waiting room. I had to sit on a table because there were no seats, and then I had to go into the waiting room for my 10 minutes with the physician. And there are millions of family caregivers around the world, in the US, everywhere else, who face the same dilemma, and it's just unnecessary if you were to build information into the system, into the treatment system itself, rather than the way we do it right now, which is this. This is the way healthcare looks to the average patient right now. It's as though the only way to build a new house was via the Home Depot. You're a do-it-yourself, some assembly required. In the healthcare industry, we sell components. We don't sell finished products. And imagine what your car would cost if you had to buy all the parts, take them home, bolt them all together. It would cost $100,000 and it would never work. And this is the dynamic that we've built because people like me follow the existing reimbursement scheme, and we target our development efforts against, you know, the existing pathway, which means we develop products that are for propeller heads. I build products historically for people like me, doctors, and the problem with a chronic disease is if you have type 2 diabetes, the end user, the family, the patient is usually not a doctor. And even if it is a doctor, they're sometimes the worst patient. So this is the system that we're working on at Proteus to help deal with this. We call it the RAISIN system. And basically, what we've spent the last 9 years working on is to network the daily pill. The idea is if it's an information problem with chronic therapy, why don't we take existing drugs and transform them into something else by building information into the drug. So imagine that you had a drug where every time you take the pill, it signals within your body what's been taken by ID code. So you get supply chain integrity, you know when it was made, you know when it was taken. Simultaneously, the monitor the patient is wearing is recording their physiologic response. So you know what the patient's doing, how their heart rate is doing, and so on and so forth. Periodically, that patch uplinks to a telephone and then to the network. And at the internet server level, you can integrate anything else in your life, any diagnostic tests, your blood sugar, your weight, your blood pressure, everything is time-stamped. And so now you have therapeutic context. So if you're a family member, you know whether to worry about mom or not. If you're a physician at the clinic visit, when you walk in and ask Mrs. Smith how she's doing and you have 5 minutes to spend with her, you already largely know how she's doing. You know if she's complying with her meds and how she's responding, and if she's having issues, are those due to noncompliance or due to a drug reaction or the wrong therapy. I'll take you through how this works in a little bit of detail here. You'll note these 3 pictures. The core of the system, and what we think anyway, is a revolutionary innovation, is the ingestible event marker. It is a grain of sand sized piece of silicon. This is 1 millimeter square in slide A, picture A, about 300 microns thick. We get it from a CMOS foundry, and we have a MEMS fab of our own, and we add mineral layers on either side of it, that it becomes powered when you swallow it. It sends a private signal conductively within your body only, so it's not RFID or RF of any kind. It's like a digital EKG. It sends a unique serial number conductively in your body when swallowed that says, hi, I'm Lipitor, I'm 10 milligrams, and I was made at such and such a place. We then integrate that into a tablet, and as you see in picture B, we can either put it on top of the tablet, which is fun for looking at, or we can compress it inside. Another vehicle we've used successfully in many clinical studies to date is in slide C, and this is an over-encapsulation vehicle, so we'll take a drug like valsartan, which is a drug for hypertension, and we'll actually co-manufacture it inside a capsule so that we can run a clinical study and actually know about therapeutic behaviors and how those are impacting the various monitored parameters that we see. This is how it works. It's really kind of cool in the sense that if you all remember the lemon and potato battery experiments you probably all did as your first engineering project back in elementary school, it's the same idea, only you're the lemon. And the idea here is that we have small amounts of dietary essential minerals, very thin film micron quantities or microgram quantities, forming an anode and cathode when swallowed. So when in an ionic solution, this system is biogalvanically powered. Just as an aside, if you ever have time and are interested, this isn't unique to us. Back in the 60s, when mercury cell pacemakers were no longer delivering the goods and people were experimenting with different power sources, including atomic pacemaker batteries, which are kind of fun. Last I checked, there are still 8 people walking around with General Atomics pacemakers from the 70s. They last a long time. But also with biogalvanic power sources like this, things that utilize the ionic environment of the body to generate power. We size these thin films to last for about 5 minutes. You swallow this, charges up a circuit, and we then have an oscillator, and we short the battery in such a way that we send a binary phase shift key signal to deliver the identifier. And we modulate the supply voltage, your body essentially, and we have this recorder on your skin that's detecting that voltage, the far field. We have this skirt. It's an ethyl cellulose disc around it, and it creates a virtual antenna, if you will. It forces a longer current path, so it creates a larger dipole, electrically speaking, which is kind of cool because the skirt is digestible, and it's very inexpensive to make, and it helps to make this very cheap. Inexpensive is really important because one of the problems with the vector of innovation in our country is that everything always costs more. So you get more in healthcare, but it always costs more. Compare and contrast that with your phone or your computer. You either get more for the same price, or you get more for less price, or you get the same amount for much less price. And this has led to some real problems in how healthcare is delivered around the world. Today, if I talk to pharma execs, typically their models show them accessing about 600 million people total in the world with their products, and everyone else just can't afford them, which I don't think is an appropriate way to go. Well, this technology leverages semiconductor economies of scale. So I can make 30,000 of these on a $1,000 wafer, and just by scaling up the process and moving to a different line width, I can get to 10 times that on the same wafer size. So I can make these things really cheap. It has to be really safe, though. So it's food-based computing. Essentially, what you're doing is you're swallowing a grain of sand, and then you have a small amount of magnesium and a copper salt, a very small amount, nanogram quantities that get into your body. So these are all items that were in your that are in your Centrum tablet. They're essential minerals in your daily diet. And one of our challenges to the design engineers was pretty much what you're looking at is the ingredient list. Being paranoid and thinking perhaps not without reason, that the universe exists partly to get me and my companies. I was very concerned about transit time and these devices getting sequestered in the gut, and so we ran years of animal experiments and tried to cause problems and damage and so on and so forth. The good news about something that we intend to go into millions of pills is that it appears to be very safe. The human body appears, unsurprisingly, to have evolved to deal with small amounts of sand really well. If you eat a salad, you go to the beach, you eat an oyster, you get sand in your diet all the time. So it appears to be quite safe, which is pretty good. Next component of the system, how am I doing for time? Next component of the system is the monitor. This is the Proteus Personal Monitor. It's a patch. It's disposable. It's designed to cost about $1 a day. It lasts for a week, then you throw it away. This has a 3-axis accelerometer on board, a thermometer, impedance monitor, biopotential amplifier, so I get heart rate variability, respiratory rate. I have your body angle, your activity, and then via Bluetooth we uplink the adherence information, the confirmation of dosing, and then at the server level we can integrate other things that we're interested in doing. This just shows the data flows, and I won't dwell on it except to show that this is personal information. It's non-RF, it's conductive mode in your body, hits the detector then has a secure Bluetooth connection to your phone, and there are 5 billion people with cell phones now in the world, and if you believe the folks at Qualcomm, in 10 years' time there will be 60 billion cellular devices on the network. Now, clearly, everyone in the world won't have 10 phones, but they will have 10 data radios in their life. And if you doubt me, just think already of what you see with your Kindle and with your iPad and with various other devices in the real world that we don't think of it as phones that are now having dedicated data connections to the ubiquitous wireless network that is everywhere. So these data then get to the cloud. Once they're at the cloud, we can associate other information about your life. You can connect to your Google Health account, to your lab results. And then, this is critical, you can share with the 50 million Americans who function as family caregivers. Family caregivers just in the United States alone spend on average $7,500 a year out of pocket. They spend an enormous amount of time caring for typically an elderly loved one, and no one in our business offers them any kind of tools. As I already described my own experience, and I'm sure all of you have had this experience or people in your family have, no one includes them in the value proposition around a product. The value proposition is aimed at an FDA reviewer and a prescribing physician, and we think that's going to change. Just very quickly, and I can get into more depth during Q&A, we have completed a lot of different clinical studies. We started with healthy volunteers, trying to try different body mass indices, trying to stress the system with different kinds of diets to see if there were failure modes that were consistent. We then moved into sick individuals who were co-ingesting these devices on placebo, tuberculosis patients in this case. We then moved into co-formulating, creating drugs with these devices in them in the heart failure population, and you'll see a slide on that in a moment, as well as a hypertension population. We've used these studies in our own employees, including me. We now have studies underway in heart failure in the United Kingdom and mental health here on the East Coast, including at Mass General right now. We're about to start a transplant rejection study with our strategic partner, Novartis, who's building this into their line of transplant immunomodification drugs. This is an engineering display. This is, on my prior companies, probably where we would have left the product, because this is the propeller head view of the world, but since we're all propeller heads here, thought I'd give you a sense for what the system can do. If you look at the top graph, that shows IEM detections. The y-axis is the identifier for each pill. The system can discriminate between multiple ingestions. If you take 5 pills at once, it will find them all. And then we have the dates along the right. You then can see step counts from activity, heart rate. We then can average in or stream in other data from other devices. In this study, we had a telemetric weight and telemetric blood pressure as well being provided. This is a clinical anecdote showing what the system is capable of. In this case, what you're looking at is, again, an engineering-oriented display from an ambulatory 82-year-old heart failure patient, class 3 heart failure, living at home. The physician was blinded to these data during the trial. What you see on top here is the patient taking a placebo labeled with our system, that's the green box, as well as the amber circles are the drug. That's furosemide, the fluid pill, the diuretic used to treat fluid accumulation in heart failure. And we can see here that from the accelerometer, body angle is an interesting dynamic. Patients, as they decompensate in heart failure, experience orthopnea, they have elevated pulmonary pressure, and that causes them to have to sleep at an elevated body angle because otherwise they feel like they're drowning. And you can see that this patient, blue, indicates I'm either standing or sitting upright, is having very disturbed sleep, sleeping at a higher angle, very agitated during the day and the evening. We see self-titration of furosemide. The patient's supposed to take 1 pill a day, and in this case takes 2, and then an hour or 2 later, another one. And then one can presume a diuresis because the patient suddenly is having no trouble at all sitting or standing upright. You've got a solid blue during the day, no blue at all during the nighttime sleep period, seems to feel fine. And then something that's pretty typical, at the end of the week, when asked by the physician how the week went, everything was fine, took my meds as directed, no problem at all. This is the #1 cause or the #1 driver of healthcare costs in America. It's the most expensive single thing CMS, Medicare, pays for is readmission for heart failure decompensation. Fully 50% of patients are readmitted within 90 days of discharge for this, only with failure to successfully self-titrate out of it. With all that said, this is what the patient gets to see, and this is something really important. At my company, we recently hired the head of connected health from IDEO, a design firm, who is focusing us all on user experience. Our goal is to delight the end user, and the end user is not me, it's not the physician. And so we've come up with a tile metaphor. Here you see it on Android phones. The idea is to use any sort of phone, have the patient see simple graphics showing how they're adhering, be able to look at how they're sleeping, click on the, or rather touch the folded corner and then see a detailed data display if that's what you're interested in. Allow third parties to connect. If you're a cystic fibrosis patient and the Children's Hospital has cystic fibrosis research going on, why can't they write an app to connect to your data and our data system? Why can't you have your Google Health tab there for the rest of your EMR? It would be a more comprehensive solution. Similarly, for physicians and providers, the notion is that you'll have an electronic chart in the future. You have limited resources to reach out and deal with your patients. Instead of blindly calling everyone, including people who are doing just fine, you should have a sense for who needs some intervention. This slide is to remind me to mention that this is the most powerful medical invention so far in the 21st century. It also represents a business model which is very successful. Instead of treating the developing world as charity, where you drop off some drugs now and then because people are poor, and I'm all for charity per se, it's much more sustainable to treat the developing world as a business. And people at Nokia, and people at RIM, people at Microsoft, at Oracle, the tech industry has figured out how to create an ecosystem of local partners and multinationals who manage to make money all around the world, and that is a sustainable way to go. Many countries outside the US, in fact, are skipping a lot of our expensive infrastructure and are really skipping straight ahead to many of these solutions that you see right here around using mobile connected health, not just what Proteus is doing, many, many other exciting things going on in terms of leveraging wireless everywhere. And again, I'll blast over that. This is not what consumer-focused means. This is, life in a bathtub is not delighting the consumer. Well, it might be delighting the consumer, but this is what many pharma people first think of when you say, gosh, you need to be more consumer-centric and reach out. The really forward-thinking ones, though, understand they need a new business model that's sustainable, that delivers outcomes, not milligrams of a chemical. This is the way your mobile phone looks for you. Compare it to the spider web I showed earlier, and this is the way your health care should be delivered as well. You pay one price, you take medicine that you need, you get the information that you need and all the rest of it for free in the background. If our technology is really advanced enough, it needs to disappear for the 80-year-old patient. And if you think of an iPhone now, I'm a very early engineer, right? I was a ham radio operator and all that kind of stuff, still am. And it's really cool how cellular telephony works, but most people in our lives don't know and they don't care. They just need it to work, and they don't need to care, which is what's really exciting. I'm going to say something kind of controversial here, but I think in the future, A song for Apple is a drug for Proteus. It's content that we can repurpose, we can give away, you can monetize people in various ways. The notion is that there are plenty of great generic drugs out there. The information wrapper, the connectivity, the system around that drug is what's required to make it realize its potential in an individual patient or family's life. And if you think about it from this perspective, which admittedly goes a little over the top, it's just a useful reframing to get you out of the traditional box of how we all think about life so far. And I'll leave you with this. 3 generations are here in this Indian family photo, which is a great thing. It wouldn't have happened in the 19th century. 20th century medicine has done some wonderful, wonderful things. But there's a lot of caregiving and information demands to make modern technology work. That's sort of embedded in this photo here that's causing a lot of stress. And the next step for medical technology, I think, is to really begin to address that. And that's all. Thank you.
Outstanding. Okay, so now our second panelist, speaking about healthcare from a different level of biology, is Professor Jim Collins. Professor Collins is the William F. Warren Distinguished Professor, and he's a professor of biomedical engineering. He's also a Howard Hughes Medical Institute investigator. He's also a founding core faculty member of the Wyss Institute for Biologically Inspired Engineering at Harvard University. There's about 1,000 other titles, but let me just talk about him a little bit. There's a whole thing listing Jim here. But Jim is a MacArthur Award winner, also known as the Genius Award. He's been at Boston University for how many years now? 20 years now. And is known to be one of the leaders in the world at the intersection of engineering and systems and synthetic biology. In fact, widely recognized as one of the pioneers, if not the primary pioneer of the field of synthetic biology, a field which takes engineering, integrates it with biology to create new kinds of biological systems for certain specific purposes. He's also one of the premier professors as an educator at this institution, having won the Metcalf Cup and Prize for Excellence in Teaching, the highest teaching award offered by Boston University. The field of synthetic biology spearheads the use of nonlinear dynamics and molecular biology to design and construct engineered gene networks, leading to the development of novel systems, as I mentioned before. I don't get into long detail on him, but his work is perpetually highlighted in the premier journals in the world. Most recently, Jim and his colleagues had a work featured on the September 2nd cover of the journal Nature, which you'll hear more about, I think, today as well. He has a degree in physics from the College of Holy Cross and a doctorate in medical engineering from the University of Oxford, and he was a Rhodes Scholar. Please join me in welcoming Jim Collins.
Okay, well, I want to thank Ken for the very nice intro and the invitation to speak to you today. Can you guys hear me? I know you're taping. Do you need me to be on the mic? Okay, give me one of these. Okay, so what I want to do is spend 20 minutes or so giving you a feel for these areas that Ken mentioned, and that is our work in synthetic biology and systems biology, but really how it impinges on this notion of personalized medicine. When the posters for the symposium went up, folks in my lab came to me and says, "Oh, Jim, is that you?" And I said, "What do you mean, is that me? That's my name and that's my face." And they says, "Well, is that a mistake then?" I says, "Why?" And he says, "We don't work on personalized medicine." And I says, "You're right." And I said, "I didn't come up with this topic. Ken came up with this topic." But I thought that Ken actually had a really good insight, because as we began thinking about what we do in our lab and what's happening in these two fields, then in fact, the fields are beginning to impinge on personalized medicine. And I'll highlight this in just a few case studies. In synthetic biology, I'll specifically talk about work on targeted antibiotics. I'll talk about work on the human microbiome and efforts now to re-engineer the microbes that make up our gut, as well as then now effort, probably that's got a lot of attention from some in the room and certainly nationwide, which is stem cell therapy. On the system side, thinking about it, realize, in fact, most of our efforts are directed toward personalized medicine, both in terms of identifying disease signatures. What's the stage of a disease in a given patient? Are they sensitive to a given drug? And can you actually use their physiological response, some of the types of responses that George was referring to that his system monitors, but now at the genomic level, can we use that to design intelligent combination therapies that can address a given individual's disease, but also their physiology? So let me just give you a little feel for this idea of synthetic biology. This is a field that's only about 10 years old. And so for the older engineers, it's really now an opportunity for bioengineers, physicists, mathematicians to play in molecular biology. So what happened was that we realized that you could go in and take motivation from electrical engineering to come up with circuits that are based on schematics that you would get in the EE department here in this building. You would then do mathematical modeling in most cases to see, does your circuit behave as you'd like? You would then go into the molecular biology space and grab components, promoters, genes, little bits of DNA, RNA, to build those circuits, get it into cells, and to see then, does it function the way that you think it would function? Just as you would, say, breadboard an electronic circuit in an EE lab, now we're breadboarding inside cells circuits that are biological and wet in nature. So with this in mind, there's been a whole host of different circuits developed by our labs and other labs around the country that involve different switches, bistable switches, multi-stable switches. They involve oscillators for periodic drug delivery, for example. They've involved RNA switches that can be used to control multiple genes simultaneously, be it in the body or in microorganisms to produce fuels or specialty chemicals. There's efforts now on developing genetic counters, circuits that can count the number of times a cell divides or the number of days that have transpired, and I'll come back to that in a few minutes, as well as genetic timers. Can you engineer a yeast so that it will now flocculate for your microbrewery exactly when you want it to drop out your yeast after you ferment? Tyrone's happy with our development on that. So better engineered beer out of BU. And then broadly, can you use these circuits to reprogram cells to do interesting and novel functions? And what I want to do is focus a bit now on that first topic, which is antibiotics. Why focus on antibiotics and what has happened? Well, the reason why you want to focus on it is captured in this very scary slide. Right? What you see on the left is that the number of resistant strains, that is, strains that are resistant to antibiotics, is going up dramatically in the last decade. Further is that these resistant strains, which used to be in the hospital, are now outside. So in the past, you used to say, look, the worst place to be sick is a hospital. If you're ever admitted to the hospital, and George can attest to that, get out as fast as you can, because there are nasty resistant bugs out in there that you don't want to get. If you get, they're going to be very hard to treat. Okay? This danger now, where they're on the outside, if you go to the gym, that's why they want you to wipe down your scheme. It's not purely for being nice to the next person. It's that there's basically MRSA, methicillin-resistant Staph aureus, that's out there. It's in the kids' child care centers. It's all around. Football teams, wrestling teams. It's a big problem. And why it's becoming bigger is captured in the far plot. And that is the number of antibiotics that are being developed is dramatically dropping. Pharma is abandoning the space. It's not a very large market, and it's a piecemeal market. And in general, you'll take antibiotics for the most part for a 2-week course. Tend to be relatively cheap. So it doesn't make economic sense for pharmaceuticals. Instead, they focus on chronic conditions like cancer and diabetes, or they focus on lifestyle drugs. So they're developing drugs for the guy in the bathtub that George showed. And in fact, a friend of mine told me a stunning fact, and that is that there are more Viagra-like drugs in development than there are antibiotics in development. Now, I'm a positive guy, so I send it positively. Some of us may die from resistant infections, but we'll have a smile on our face. So, with that in mind, what can you do? And what is synthetic biology doing? Well, in our lab and now other labs, what we've been focusing on is re-engineering organisms to go after resistant bugs. And what we've been doing is focusing on re-engineering bacteriophage. It's a weird-sounding organism. It's basically a virus that only attacks bacteria. So what these things will do is they basically be like a little lunar module. They'll dock on a bacterium, blow their DNA in some cases make multiple copies of themselves and burst the bacterium. In other cases, they'll actually harness the machinery of the cell to make additional proteins. Tim Liu in our lab, who's now an MIT faculty member, went in and engineered them to actually produce proteins that when delivered in conjunction with an antibiotic could make the antibiotic much stronger. Okay? And that's one of the things you want to be able to do. You want to be able to take lower dosages of antibiotics. 1. 2 is he found that he could use these to resensitize resistant bugs to drugs that they had become resistant to. Now, what was interesting about the development in this space is when we first got involved in antibiotics, and those in the pharma can attest this, even just 5, 6 years ago, the charge was you needed to be broad spectrum. What does that mean? That means you need to develop antibiotics that will kill everything. That's the goal. Can you kill everything? What's really happened in the last 3 or 4 years is that's changed. It's changed for the next slide that I'll go in, and that is we've now appreciated that we're populated by a lot of microbes, and you don't want your antibiotics just wiping out your natural flora. So it's flipped, whereas in the past they wanted broad spectrum. Now everybody's talking about doing very specific directed targeted therapy that's on a personalized level. And it turns out phage, which have been panned, and I can talk about that in the panel, for many different reasons, one of which is that they're highly specific. So bacteriophage are specific to certain species of bacteria, and in some cases specific to strains. So in the past, commercial guys ran from it and says, how are we going to have that? Now it's being embraced. Say, okay, if we can now identify that you have infection A by this bug, wow, if we can target just that guy and wipe it out, we can now save your flora. And what's interesting on this is this technology has been spun out of BU into a company called Novophage. Mike Kerr is a former PhD student in my lab, is one of the guys running it. Tim Lou's an advisor, I'm an advisor. We got contacted by the US Army on the basis of this technology, and we're now working with the Walter Reed Medical Institute and the US Army to help returning US veterans coming from Iraq and Afghanistan. Turns out these poor characters are coming back with very nasty wounds. They're infected by 3 different strains of bacteria that we don't have good drugs to treat. But it turns out those bacteria actually have bacteriophage you can go after. And so we're now working with them to use this technology to go in to specifically target and help out some of these soldiers. This second space that I've already talked about is this idea of the human microbiome. It turns out that we have 10 to 100 times more bacterial cells in our body than we do mammalian cells. Okay, it doesn't mean you're a bunch of bacteria. You know, I knew there was something about Ken that bugged me. But it's not that you're predominantly bacteria, right? It still only constitutes about 3 or 4% of your mass. But we're beginning to appreciate more and more that there are hundreds, close to 1,000 different species of bacteria making up our bodies, contributing for the most part in very meaningful ways. But we're also beginning to appreciate that the microbiome will get altered. It'll get altered as a result of genetics. It'll get altered as a result of antibiotics. It'll get altered the way what you're eating, what happens to certain diseases. And that will throw things out of balance. And more so, folks are now identifying allergies linked to altered microbiome. They're looking at chronic conditions like Crohn's, different metabolic conditions, even obesity, as basically linked to modified microbiomes. And synthetic biology is stepping up on this, say, okay, can we now go in and re-engineer the bacteria in your guts to now express proteins that you're lacking? Can we use bistable switches that you can go in and take one of George's pills, flip it on, and now allow these bugs, these re-engineered bugs that you have inside your body to produce stuff that you need? We're using phage as one of the delivery mechanisms. We're also going further, and Jeremiah Faith, a former PhD student of mine, is now at Washington U. He's actually looking at creating synthetic ecosystems. So can you go in and now repopulate somebody with 10 microbes that they're lacking? So say if you've wiped out most of your flora due to antibiotics or had a condition that wiped it out, could you now predict how you could repopulate creating this synthetic ecosystem? And he's making very, very nice progress. Okay, so you would think this is great. Synthetic biology is fun. We're a bunch of propeller heads. All of us must love it. The public must be embracing it. Well, that's not the case. Ken wanted me to briefly mention ethical societal concerns. It turns out there are a lot of people pretty scared about synthetic biology. And what are their concerns? Well, number one concern that comes out is that the engineers are running amok in molecular biology labs. They don't like us in there. They think we're tinkering with life. What you'll see, and I'll make reference to why they're worried about that, they're worried that we're going to construct a Franken microbe so that something's going to escape from my lab and gobble up City Hall or worse, Fenway Park. Big, big concerns. And part of it's arising from my colleagues that are speaking irresponsibly about where the field is and hyping up possibilities as where we are. Right now, we're basically genetic engineering on steroids. The field is very early. Not a lot is happening beyond kind of small lab baby steps. What caught the biggest attention, and for those of you who probably first became familiar with the topic, was when Craig Venter, my colleague, about 6 months ago, announced that he had created so-called synthetic life. He didn't, and I'm happy to explain why I don't think he did. He did a major technological advance, but it caught the cover of The Economist that man has now made life, and we haven't even come close to making life. We don't know enough. But it's now caused a fair amount of concern, and is an appreciation, I think, increasingly from Ken's leadership at the college about creating societal engineers, engineers that want to impact the communities, our communities positively, but also to educate the public about the misuse and the proper use of technology, including biotechnology. And so we have a long way to go. So most of synthetic biology is in microbes, playing around with bacteria, playing around with yeast. Much of it is moving toward biotech, industrial biotech. So the challenge is for human health, okay, fine, we're made up with a lot of bacteria. We do have some yeast. Most are good. Could you now move up the food chain? Could you move to higher organisms? So can synthetic biology play a role for our dean? Yes, I could draw the direct evolutionary link there if I had a little more time. So there is now a growing number of labs working in mammalian synthetic biology, one of which is ours. And there's a lot of exciting space there. The most probably interesting and exciting areas is in stem cells. So there's been a lot of controversy about embryonic stem cells. I'm actually not going to touch upon that, but touch upon the other area, which has alleviated some of the concern, and that's this notion of induced pluripotent stem cells. That is the idea you can take adult cells that have already differentiated, so take human somatic cells, deliver 3 to 5 different transcription factors, often known as the Yamanaka factors. So Yamanaka was a Japanese scientist who discovered this about 4 years ago. George Daly, our colleague at Children's Hospital, was a kind of a co-discoverer and similar kind of pioneer in this space. But the idea is you can express these, and in a period of time, you can actually reprogram these into so-called induced pluripotent stem cells. And that is now these are adult stem cells that have embryonic-like properties where they can be redifferentiated into cells of interest or tissues of interest. So the idea would be that you could take these out of a patient, make them into patient-specific adult stem cells that now could be redifferentiated into a tissue that's needed. Either it's your skin, or if you lost your liver, or have tissue damage, need for heart valve. Very promising. One of the challenges is that while this sounds great, in fact, it takes a long time, about 8 weeks in general, to begin to get induced pluripotent stem cells, and the yield is remarkably low. Less than 1% of the cells will convert in that period of time. What we did from a bioengineering standpoint with George Daly and Derek Rossi, and work we just published, it's just online now, it's going to come out, I think, officially next week, is that we came up with a new technique that now, instead of using viral vectors, so in the past you would need to infect the cells with a virus that would carry the transcription factors, which carries a number of cancer risks and other risks of altering the physiology, is that the team came in, Hu Li and others in my lab, working with the folks over at Harvard, used synthetic biology notions where they created synthetic mRNA that they could deliver directly to the cells for reprogramming. And what they were able to show is that they could dramatically drop down the time for the reprogramming and increase the yield by 100-fold. And so now you no longer have the risk that the cells that you made into these kind of new adult stem cells would become cancerous. That basically is now eliminated or largely reduced because you don't have the viral problems. Second area is that work prior to stuff I'll just briefly mention here was requiring somatic cells. So you'd need to kind of pull skin out of Ken or whoever needed them and then do it. And it'd be painful, et cetera, for different biopsies. With John Low and, again, Hu Li and George Daly, we use bioengineering techniques to now come up with approaches where you can just take from peripheral blood. Take cells from peripheral blood, draw from your patient who needs the help, and from that now, using those early techniques, very quickly get to stem cells that can be redifferentiated. So there you're going to hear a lot about, not per se our work, but this as a therapeutic space in the next decade, 2 decades. We're still a long ways off, right? So you've got this initial stage where you can use either the embryonic stem cells or these adult stem cells. The challenge still from a bioengineering standpoint is how do you get them into the types of tissue types that you want, and then how do you actually interface it with the patient appropriately. So there's beautiful, marvelous opportunities for all of us in this room to get into this space, and I suspect that at least some of the young ones will probably see some therapies in their lifetimes taking advantage of these. Very quickly, I want to just get into this last topic of systems biology, and it impinges some of the great stuff George talked about, and how this links to personalized medicine. So our group and other groups in the engineering space realized a few years ago that instead of just kind of putting together circuits as an engineer would, that we could use those circuits and other techniques to figure out how a cell is wired up. Right? So for us engineers, you either would make the circuit that would make the radio or make the iPhone, or you would take apart the iPhone to figure out how it's wired up, or take apart your radio. And we developed systems biology techniques that enabled us to go in, turning different components of the cell up or down or on or off as a means to infer how they're wired up. Which genes are talking to which genes, what's the nature of the interaction, is it exciting the gene downstream, is it inhibiting the gene downstream, and how strong is that interaction? What we realized as an engineering group just about, again, 8 years ago, was that you could, once you got that quantitative network worked out, that could be on the order of 10 genes, it could be on the order of thousands of genes, and we've done this both for bacteria, yeast, and human cells, that you could actually use this to identify the mode of action of drugs. So we're in a so-called target-rich age. So for those of you in biotech or read about biotech, you'll hear about targets. Targets are basically proteins that would identify that if you can knock it down, you would get possibly an interesting therapeutic effect. Biotech and pharmaceutical companies are really good at setting up chemical assays to see, does the compound under development hit the target that it identified? What they can't tell you is what else does it hit, right? So in the case of humans, you've got 30,000 genetic possibilities, probably hundreds of thousands of protein possibilities. It's the reason why, if you've now invested, your stock turns red, right? Oh, the drug failed phase 1. Oh, the drug failed phase 2. Oh, the drug failed phase 3. Oh, the heart drug, has to be pulled from the market. Why? Because you basically have off-target effects that mess you up. And what we realized from the systems biology standpoint was if you've got your network, you can actually use it to identify the actions of drugs. So I've got several of my former students from BE 402 and from first lecture and throughout the course of control theory that I teach to our undergrads, I emphasize to them, you got 3 parts to a control system: the input, the output, the system itself. Right? You know 2 of the 3, you can work out the 3rd. So in fact, we measure our inputs, we measure outputs. From that, we can infer the system or the transfer function for your mammalian cell. What we recognized then was if you've got a drug under development that you don't know what it hits, but you can measure the output, measuring how all the cells of the proteins respond, you can take that output, filter it through your network, and actually predict what that drug hits. And we've now done this to gain insight into the development of drugs, both for antibiotics, antifungal, and mammalian schemes. But we also realized from the personalized medicine side was that you could use this to identify the disease state of a patient. So the perturbation, this approach is independent of perturbation. It could be a drug, it could be a biological state, it could be the disease state of a patient. So what we've been able to show is you can take, for example, men with different levels of prostate cancer, take their measures, filter it through a network for human disease, and actually identify, this person is fine. This person has a type of cancer you don't have to worry about. This person has a type of cancer that's recurrent, maybe worry. This person has a type of cancer that's really bad, and you got to get it out. My wife's a primary care physician at Mass General whose research, among other things, focuses on prostate cancer. And she often tells me that the big challenge is the PSA test, which is the one commonly used, is highly nonspecific. You get a high PSA, but it's not going to differentiate between the kitty cat cancer that you can leave, you're fine, versus the nasty one that you need to get out. Right? And it turns out, as she likes to say, it's kind of harkens something that George said, but that the majority of men will die with prostate cancer and not of prostate cancer. And so if they think, I got cancer, get this out of me, but they don't know if it's a kitty cat or tiger, it turns out that the procedures to get it out actually have lifestyle complications on a very personal basis. And so the systems biology approaches have actually now enabled us to identify markers very quickly to help give you indication, is it a kitty cat or a tiger? Last example is this idea of now getting after drug sensitivities, but also combination therapies. So with our group, Jim Costello and Diego Camacho, they kind of appreciated that if any of you have ever looked at kind of mammalian genomic data, it is overwhelming in its complexity. We're engineers, so we embrace complexity, we live for it, right? And what these guys were able to do is say, okay, let's simplify it. Can we project the genes onto pathways and look at it in a much simpler scheme? Can we now work out transfer functions for a given patient or for a set of patients of how they respond to a particular drug? Will they respond appropriately? And can you use that to get after personalized drug combos? So what they focused on was tamoxifen. Tamoxifen is a common breast cancer drug. Looking at, in this case, this was cell culture, so with so many set of patients. But here's the response of tamoxifen. And their goal here was to say, looking at a database including over 1,000 drugs, could they find another combination of drugs that would achieve a similar physiological response? Let's assume there are many patients who are not responsive to tamoxifen. And so what they did was they were able to use a bioengineering approach to very quickly scan through those and came out with that basically a statin, which you would have never thought you'd want to give for breast cancer, by itself wasn't doing anything interesting. So if you compare the two now, these are a small subset of pathways. Red is that they were upregulated, blue is they're downregulated. That doesn't look so much like anything's happening. When they then found another drug, Benflurafex, I don't know actually what indication it's prescribed for, that by itself, a little closer to tamoxifen, but still not so much. But when you combine the two, it actually looks remarkably like tamoxifen. And this was one of the combinations out of the 1,000 they looked at that actually gave an effect that looked quite similar. And when they actually went in experimentally, they found on these two drugs that previously would never have any indication for breast cancer could, in fact, begin to kill breast cancer cells, similarly to how tamoxifen would. And I was thinking through George's talk as he was giving, we're also thinking about how you could use this to examine what are the potential consequences of taking N number of drugs as so many patients do physiologically on the patient, right? We don't have a lot of data on that. And so the notion would be, what are the side effects that 80-year-old Mrs. McGillicuddy is going to get because she's now added drug 7 to the 6 that she was taking? And this type of very rapid personalized analysis can very quickly identify, oh yeah, you don't want to do that because now she's going to start acting like a college dean. Okay, so with that, I'll end, and this is just our website that I encourage you to check out, and I think we'll now just turn to the panel, I suspect.
Thank you, Jim. So we've heard now from two pioneers in this world of emerging personalized medicine, from the biologics and drug design to the technologies that will fundamentally alter the way we interact with the pharmaceutical world and the medical healthcare system. I open up now to the audience for questions, thoughts, comments that the panelists can respond to. Certainly somebody out here has something. Right in the back.
Well, what would be potential risks for engineering our own microbes? What could be the potential risks to us?
So that's an interesting question. So a potential risk would be the kind of unknowable, and that is that can you appropriately predict what will happen when you put in an artificial genetic construct into your cells? You know, you'd want to minimize the potential adverse effects, so you want to be careful on how you deliver. And I'll bring up an interesting example. So phage are known to at times trigger the sharing of plasmids between cells. So phage are all around nature. They're very common. And when they infect a bacterium, they can trigger a response where the bacteria will now want to share other bits of their DNA with bacteria around them. Where that could become a problem, and we need to explore to make sure it wouldn't in this case, is that you wouldn't want bacteria sharing resistant genes to your treatments widely, either in a person or you're going to kind of have this material generally shared unintentionally between people in a house or in a community. And so that is one potential worry for this kind of technology, particularly on the way that we're considering delivering it.
Hi guys, fascinating talk. I'm a double E, and I've never really been exposed to what you guys have been doing in biomedical and synthetic engineering. So the question though is that in IT and in computing we have Moore's law that really does a great job in predicting performance and cost. Healthcare industry doesn't have anything at all like that. The work that you guys are doing, can we basically get on a curve where we have medicines and treatments that have the advancements that we've been seeing in the computing industry for like the last 30 years?
Well, I certainly think so, and I'm not alone in this respect. I'm blanking on the author's name right now, but there are other authors that have really looked at the productivity of healthcare, and the fact that by 40 years ago or so in the United States, we set in place the current cost reimbursement system, largely driven by Medicare, and that set up a certain set of assumptions and presumption, which is that we invest in absolute therapeutic capability. If you can't treat a disease, I'll pay you a lot of money if you can. And if you come up with a better treatment, in theory, according to this kind of certain set of testing, then I will pay you a lot more for that. What has been less rewarded has been the periods of consolidation that most innovative industries find where, look, we have plenty of absolute therapeutic capability out there, and the best return on investment could be made in making the therapeutic capability we already have work better and more efficiently for more people. And that means applying, I think, in this case, Moore's Law type information technologies to these therapies we develop. Most things that are brand new are very expensive. It's that way in IT as well. The brand new chip, the brand new computer, the brand new architecture, it's expensive, and then they spend a lot of time really wringing the cost out.
So let me touch on this also. So I think that we are seeing Moore's Law in certain biotechnologies being related to Moore's Law, so things such as DNA sequencing, DNA synthesis. Now you're seeing the log linear relationships that are similar to Moore's Law. But I think you touch upon an interesting point that I think the technology, it's fine, and we're seeing that. I think on the therapies, I haven't seen the corresponding shift. And I think part of it is that the therapies require biological insight or medical insight. And I think we as a community have made the false assumption that information will correlate very nicely with insight. And so you look, we're about 10 years into the human genome sequencing efforts being finalized. And people are very disappointed. So they thought, okay, we sequence everything, boom, we're all going to live forever and we'll be healthy. And it hasn't happened. I think it hasn't happened because you've got this mass amounts of data, but it hasn't given us a lot of mechanistic insight in what's going on. And so I think our understanding of how the body works and how that understanding can be translated into new therapies is still very low throughput. And I think so Ray Kurzweil is one of the authors who you'll hear talking now about biology as a technology, and I don't think we're there, and I'd be surprised that we get there anytime soon, given how hard it is to get real insight.
Yeah. People didn't specify themselves. That's the problem.
So on the personalized medicine front, I wonder, in your opinion, how far away are we from getting molecular information from a patient about their disease and then prescribing some form of treatment with significant improvement on the state of the art today? And assuming we're not there yet, what do you think is the biggest hurdle right now to that technology?
So I think you're going to soon see biotechnology in the doctor's office for relatively easy diagnostics. So I think that there are a number of biotech companies that are moving toward using biomarkers for relatively straightforward diagnostics. I think we're a ways away from, in many cases, having the information translate into dramatic improvements in therapy, to kind of paraphrase what you mentioned. And again, I think it's because there's still this gap that we've got all this marvelous info that's beginning to be generated, new capabilities to kind of make the link, but there still is this now additional gap between biology and chemistry. So if you talk to pharma companies, they will have a relatively small biology effort and a very big chemistry effort. And most of what we were talking about, and I think much of where the disease is on the biology side. And so there's still this gap on that if I've got this sort of profile, what is the best chemical treatment, which is what just a drug is, that would enable me to reverse what I see. And there I think we're still a good ways away.
Is it validation that's necessary?
I think it's validation. I think it's, some of the stuff I presented and what my colleagues are doing is still very, very new, very young. There is also the gap in terms of going from these projects to actual products. And I know our college and others are working hard to try to close that gap, but in many cases, it's still so far out that companies are increasingly, in these tough economic times, understandably, needing to focus on short-term revenue. And pharma is not doing so much in R&D right now.
The only thing I'd add to that is the dimension of the regulatory system not being equipped by statute or by experience for handling this. And so if you think about the regulatory superstructure in the EU and the US, for example, they were primarily developed around traditional categories of drugs, then devices, now biologics. You come up with these very specific targeted combinations, and there's no good way to fit it through except by saying, that billion-dollar process it takes to approve the everyone's in a bathtub drug that people take forever? We're applying that to this phage drug that will only work against a certain strain that you only take for a couple of weeks, that will, and without some creativity on the regulatory process, which really has to come from lawyers who are legislators, it's going to be very difficult to make the leap economically.
I think maybe just pick that up real fast. I think one area where you're going to see it, the diagnostic and then the improvement in care, probably one of the quicker areas is in diagnosing bacterial infections. So if you have a bacterial infection, you go in and they're going to make a guess on what you have. And they're going to wallop you with broad spectrum antibiotics to try to get what you have. And in some cases that's going to help, in some cases not going to help. And they don't have a lot of time, and the trouble is that our current diagnostics take often several days to get a readout on, okay, what is the strain or the species in there? Is it drug sensitive or drug resistant? And there are efforts at the Broad Institute and coupled here with BU to actually try to develop basically a box that can be in a clinical unit or a doctor's office that can, within minutes, identify what's in the patient and identify as a drug sensitive, drug resistant. It's, you know, it's still talking years away. So you'll see slides on it within probably 2 years, but I think you're still many years away from getting it cleared regulatory-wise. But I think that'll be one of the first ones you'll see.
So since I'm holding a microphone, I want to, speaking of lawyers and things that are closer by, George, the technology you talked about is a fundamental paradigm shift potentially in the role that the patient and their immediate family plays in monitoring and adjusting their health care regimen. Not just compliance, that's a different issue.
Mm-hmm.
And there's a lot of potential for the patient or family or whatever to not abuse it, but misuse it and get into trouble, perhaps. You know, and then suddenly say, well, I thought I was following directions. Nobody gave me particular directions. I'm suing you. Because why didn't the doctor, you know, I thought I was supposed to take more pills based on this readout, so on and so forth. So how is this technology you're developing? What are the barriers with regard to the whole dynamic of the individualized person's responsibility now for their own care?
Okay. You raise a very good point, which is any kind of system can break down. The first thing is to try to avoid, from our perspective, use cases that would lend themselves to that. So you may have seen on the slide we have 99.3% detection accuracy in our latest clinical series. Our experts tell us that for the kind of trend analysis we're focusing on, anything over 90% would be adequate. Clearly 100% is what we'd like to see. You would never want to, say, remind an Alzheimer's patient, take your med now because we haven't detected your pill, and you know, have some bug in the system lead to the paramedics finding them dead on the floor having taken 20 pills repetitively. So you wouldn't want to get into that kind of reminder system. I think where the system properly has a role is by sharing information about physiology, about behaviors, feeding that back, and very critically taking people where they are and educating them over time. One of the amazing things about coming to engineering school, or I reflect on my experience at medical school, is a lot of life is vocabulary, and on your first day you don't understand anything that's going on, and by the time you walk out you kind of, you know, can follow a conversation and it all seems very familiar. Part of our problem with one-size-fits-all behavior modification and education for someone with diabetes is if I get diabetes and someone is sending me messages about my blood sugar is important and this glycosylated hemoglobin is an important indicator, well, I will be bored. That isn't appropriate for me. But I may need help with my activity or something else specific. Similarly, someone with far less education needs something more basic than glycosylated hemoglobin. And if you know something about someone's specific behavior and you can target the next intervention or interaction based on where they are in a very bite-sized chunk, you can move people over time. I liken it that you can cram for biology exams, at least I could, and but you can't cram for a foreign language fluency test. I mean, you have to do a little bit every day. And behavior modification is like that. A lot of chronic disease management is behavior.
Well, I have a question about the information sharing a little bit, because as we all notice, the technology is really getting ahead of itself. However, the privacy rights not as much. Mm-hmm. So there is a little bit of concern when, you know, involve us actually swallowing something, and then as I notice, the data kind of rotating around between Yahoo and especially Google, which is not known exactly for privacy protection and so forth. And this is very sensitive information.
Right.
And actually the company is making money from selling information about us. So how do you guys addressing this issue?
Well, you bring up a critical issue, and one that is something we think about all the time, which is privacy. And people are actually quite willing to give up parts of their privacy provided that they understand what they're giving up and what they're getting in return.
Can they understand that?
Well, and that's the point. And so one needs to be very careful about that, but I'll give some examples. People often sell their privacy for not very much. When you go to the local food store, you quite often use a reward card, which means that people there can figure out everything that's going on in your house. all to save a few dollars on your grocery bill. The most invasive privacy technology in modern life isn't the federal government or the National Security Agency. It's people like these folks. How many people here use credit cards? A lot of you? I mean, this came home to me recently. I was at the Advanced Neuromodulation, American Neuromodulation Society meeting in Las Vegas, listening to talks on, anyway, stroke rehab and that kind of thing. And it's 10 o'clock at night. We finished the last session, and I go into the deli off the casino to get my $10 sandwich and a beer to go back to my room and have dinner. And my charge is denied because someone in Visa Land had a computer alert occur that Dr. Savage does not go to casinos in Las Vegas to. So, and I have to spend 10 minutes on the phone while I'm tired down there in the deli proving who I am so that I can buy my. The critical thing, and the thing that. Yeah, exactly. The thing Visa does very well that we're taking consultants on board for and what have you is that they're very good about what they do with the data. And just think through all the political scandals you've heard of where different government records get released about your least favorite politician or something like that. Unless somebody had physical access to the copy of the credit card statement, I am unaware of any security breaches from Amex or Visa or MasterCard where somebody within the bank leaked somebody's credit card records. That's the kind of privacy we have to design in. We believe that the patient needs to own the data. And so the data is not owned by the physician. The data are owned by the patient, and the patient decides who they share it with. You don't want to share it with your doctor. You don't want to share it with your mother. Fine. You want to be antisocial. You don't want to do this technology. Fine. This isn't about control. It's a bit like a Hollywood actor hires a personal trainer to get him in shape for his He-Man role in 6 months' time. And some former Marine wakes him up at 5 in the morning to make him go running, and he grumbles. And, you know, is that Marine controlling him? Well, no, he's paying him a lot of money, actually, but he's helping motivate him to do something he really wants to do, but is having trouble doing.
Yes, but however, I'm having trouble controlling my data. How do we even know what is rotating out there? Using our cell phones, I suppose. Yep. The BlackBerry is kind of trying to hold on to some aspect of privacy. Is there somewhere that we can actually check what goes where? Where is that ownership of our information? And by the way, congratulations on the great research, because that sounds like it will be life-saving. However, how do you guys address that issue?
Well, it's a great point. What you need to do is you need to earn trust, and you need to create systems. Systems are fallible. People are fallible. You need to identify a contract people can understand. Be upfront about what de-identified data or what identified data you're going to use to advance medical science or make money or whatever you're going to do. And you need to appropriately sell that to people, and that's a case-by-case basis. There are a lot of laws to comply with. There are a lot of different companies going on. Right now, I'm actually recruiting a director of patient contract, is what we call it, because we don't view privacy as an idea of a defensive crouch, something to hide from, you know, to comply with the law, and that's all. But we view this as a positive, because if you get out there and you define the space as you describe in a positive way about, look, here's how we protect your data, and here's how we use your data, and this is why we think it's good for you. If I convince people of that, and we actually live up to our stated ethics and goals over time, that will be rewarded.
So let me, again, we have to transition now. We have a reception waiting outside, but I have to ask you to still stay seated while we give the alumni awards. Let me thank our panelists again for the wonderful talks and their insights about this incredibly important issue.